schedule.
Okay,
Yes.
started recording. Okay, you ready? Okay,
Yeah, let's kick
take
off.
it away,
Um,
Russ.
yeah, so welcome
Ready, let's
everybody.
go.
We're really excited to host this family meeting this evening. I think you're going to see that there are some really great updates, and you're going to hear a review of what we have been doing during 2024 and some of the ambition into 2025. Just a little piece of housekeeping. If you're not speaking, I would say stay on mute just to keep the call clear. And if anybody is having any issues with the translation service, just drop a message into the chat and we will assist you in the background. So we have a packed agenda. We're going to talk a little bit about our global community. We're going to give an intro into the Rory Bell Foundation board for those that may not be familiar, and talk a little bit about our purpose, promise, and a research roadmap. We're going to talk about 2024 highlights. It was a big year last year. And then Rachel and Kate are going to take you through some science basics. We're going to talk about our grant awardees, and then it kind of turns over to you guys as the audience to we really want to try and drive some engagement within the community, and it's very, very important that we have all of our families registered on RareX, and we'll talk about what that means and some of the data that we're seeing, and we'll talk about some of the non-research related goals that we have this year. It has been a very, very busy 2024 and 2023. And actually, when I was preparing for this myself, I had a quick look. I think it was April 2022 that the domain name, the rorybellfoundation.org, was registered. And when you think that's less than 3 years ago, bear that in mind as you get the updates from people as we go through this evening, and you'll see the pace and the growth that's happening in the organization, which we're really excited about. And we are going to obviously finish with some questions and answers. If there are any important questions as we go through, please do raise your hand and we will jump across to you. But it may be best for some of the questions to hold them towards the end, just to make sure that we can get through the content. We are in a really great position that we have 27 slides, which shows that we have lots to share. So without further ado, let's move forward. So Terry, if you can. So just to give an update on our global community, we have about 90 cases across 16 countries of NARS1 now identified, 15 new families in 2024 into our Facebook community. And our community's helped to raise the guts of $500,000 for research since 2022. Within our community, just to recognize that there are three populations that we work with. And again, that adds to maybe some of the complexity of what we do, but this is really just an overview slide so we can identify where in the world our other families similar to ourselves are. So we'll keep moving, Terry. So we'll do a round robin. I don't know if everybody has joined the call. So I'll start with myself. I'm Ross McCarthy. I am your Director for Patient and Family Engagement here at the Foundation. Terry, do you want to jump in next?
Yeah, Terry DeClerc. So Rachel and I co-founded the organization almost 3 years ago. I have a 6-year-old son named Grady. And so I recently we made official titles and jobs, so helping with development, but also kind of the chair of the board just to help general things keep running every day. Kit, do you want to go?
Absolutely. Hi, all. My name is Catherine, or Kit. KIT is what I usually go by. I jumped around and did a couple of different jobs at the Rory Bell Foundation since starting. I'm a longtime friend and coworker of Rachel, so when she asked me to join, I was very honored to do so. But recently, with grant money that we have coming in, which we will talk about later, it's been my great honor for the board to have okayed me as a executive director in a part-time paid capacity for the Rory Bell Foundation. So I'm super, super excited and honored by that work, and I'm looking forward to what we can do.
I see we have Rachel.
Rachel, you want to introduce yourself quick?
Sure. Yep. Sorry, I'm running late. I've had 6 hours of meetings today. I'm Rachel Hammond. I'm one of the co-founders of the Rory Bell Foundation. I think almost everybody I've interacted with probably on this call at some point. Rory was my daughter, who was diagnosed in 2020 with NARS1, and she passed away in 2021 from NARS1. So this organization is to extend her legacy and for us to also be able to help all these kids live the best lives that they can.
Thanks, Rachel. Chris, do you want to jump in?
Yes. Hi, everybody. I'm Chris DeClerc. I'm Terry's husband and father to Grady, a child with NARS1. I'm the treasurer of the foundation, so work behind the scenes with all the finances.
Thanks, Chris. And Lauren.
Hi, everyone. I'm Lauren Anderson. I joined the board last year as secretary. I have a brother with NARS1 who's on the older end of the spectrum and will be 33 this week.
Thanks, Lauren. So that gives you a flavor for the board and everybody who's put work into preparing for today and everything else that we do. So why do we exist? And Rachel, I don't want to talk on your behalf, but I guess you put this slide together. But ultimately, you know, the reason that the foundation exists is to care, champion, and change. We have a purpose, which is to champion the opportunities and change outcomes for everyone that's impacted by NARS1. And our promise is, you know, joy today and hope for the future for every NARS1 family. And we hope that that resonates with all of our families through care, through championing, and through change. We all deeply care about NARS1. We're all personally affected. I know the others mentioned their children, and I must mention Kian as well, our little boy who's 5. So we care about the gene, we care about the disease, we care about the families, and we use every opportunity we can to champion for them and to drive change. And you're going to see that, I think, in spades, you know, as we go through the next number of slides here.
Hi. Hi.
Hello.
Hi.
My name is Jasmine.
So Rachel, I think we're going to move on to your section. Talk about some research.
Yes. This is really one of our pillars for us to keep track of what we're doing and how we're doing it and how we can get to, you know, our end goal. This is something that I developed and Kit made prettier. So, and to really outlines all the things that we need to do to get to, you know, therapeutics and to clinical trials. And some of the things we are really doing a great job of with the check marks there, and some things we haven't really started, which is the X's, and some of them we might not even need because we are such a small community. And so when you're a small community, part of that is that you really are more in charge of your destiny than when you're a much larger one, and where you have to rely on a lot of external relationships and things to build up. But we've made a ton of progress in a really quick amount of time. So in just over 2 years, especially in being able to build our research toolkit up with different models and bringing together these awesome researchers that we have on this call as well, who have worked so collaboratively over the last couple of years to really move forward the direction of research. Do you
Any
have control?
questions on this? I do. Yep. Or I do.
Okay.
Okay.
It's also on our website, so you can always ask questions if you want to just keep looking at it too. Terry, did you want me to do this too?
Sure.
So this was just our highlights from last year for 2024. We actually just finished our impact report as a board, and that is also posted on the website, and we'll also, I think, be sending out to our listserv as well. That really talks about what we've done as a foundation. A big, huge win for us is what's called the CZI grant. CZI stands for the Chan Zuckerberg Initiative. So Facebook founder, Mark Zuckerberg and his wife, they gave out 30 grants last year to nonprofit organizations like us who are fighting for rare disease and therapeutic choices and clinical trials. It's $800,000 over 5 years. We cannot use it for research, but really the intention is that we're around. We're around as an organization. We're around, as a foundation to continue to help to support families, to continue to help investing in research. And so we're moving very fast with their help and have access to a lot of tools and resources. The rest that this is just the rest of the things that we've done, just a huge amount of engagement from you all in our community, grant funding. We funded
if we get a 10
a drug repurposing study through Perella Labs with money that we raised to the gala. And yeah, we're trying to just show up everywhere so we can bring a little bit more awareness to NARS1. Do we have an and? Next slide thing.
Yep.
Great, Stephanie. I'm just going to comment on this gene review manuscript. This is something that I talked with Stephanie about maybe 6 months ago. Gene reviews is a huge win. This is something that clinicians, when you get a diagnosis, and genetic counselor reference first. It's a summary, basically, of the disorder. And we have not had enough information for this to be published. And so the fact that this is being submitted and open is like a big win for our community, because it means that we'll now have information at the fingertips of clinicians who, as I'm sure everybody in this call knows, very few clinicians have seen more than a single NARS1 patient through that diagnosis. So thank you, Stephanie, for doing that. That was still on my checklist of things. This was our big thing, and why we invited Ingrid and Ilka and Stephanie to join us. We awarded $130,000 in research grants across these three lovely, intelligent women to further the understanding of NARS1. And I think I have all the slides built in except for maybe Stephanie's. Who's first? Ilka. Oh, wait, we're not doing that yet. Sorry, I don't have my slides memorized. I wanted to do a brief intro to science for NARS1 before we let them talk, so that you can kind of understand, especially for our first-time community members. You can kind of understand a little bit more of what they are talking about in relationship to what's happening on a cellular level. So NARS1, the N stands for asparagine or asparagine, which is an amino acid. We have to have amino acids for life. They build up and link each other like you're building a house of Legos, and they all connect and make these proteins that we need. I think you must have already talked about the intro in terms of how different variants are showing up in patients and what
No, we
those
did
are
not.
looking.
We
We
did
touched
not.
on very, very briefly to say there are sort of three main,
Yep.
but kept it right over that sky high level, Rachel.
Yep. Yeah. So it's this de novo. So in this figure B, the de novo or the spontaneous. So this does not inherited. So we have a lot of our newly diagnosed individuals are fitting into this sort of class. And we are also seeing this specific variant, this P, which is protein, ARG, which is arginine, and then 534, and that star means it's nonsense. So it stops making that protein. And then our other bucket is this inherited, where, mom and dad each carry a variant on their NARS1 gene, and that gets passed down. And then we're also seeing now CMT, which is peripheral. And so I don't know if I have any Charcot-Marie-Tooth families on here, but that is something that's usually happening later in life, and we still don't quite understand all of it yet. It's just emerging, but that is just peripheral, meaning they don't have as much of the global developmental delay and other issues that we're seeing in the rest of our kids. So this is really sciencey. And then the next slide, if you want to go, is it broken down as a construction site. So this is really like what is going on. Our genetic counseling student last year made this for us in a brochure. And this is kind of showing, how the protein, how NARS1 protein is supposed to be made and how it's supposed to be working in our body. And so this tRNA synthetase worker is delivering that materials, delivering asparagine onto the truck. The truck is driving over to that construction site where it's going to keep on building the blocks, building the infrastructure for your building. And what's happening in individuals with NARS1 is that asparagine, that delivery truck, is not making it to the construction site, or at least not making it to the construction site on a regular basis. Or it's missed, or it's causing some irregularities in how that building is being built. So that's what we're seeing. That is as basic as I can get it. And now we're going to move into our grants. And we're going to start off with Ilka Heinemann from Western University to talk about her research, her area of expertise, and what she's looking at for NARS1.
Thanks, Rachel. I'm Ilka Heinemann. I'm an associate professor of biochemistry in London, Ontario. So not the real London, we're just the Canadian London. So we're kind of sort of between Detroit and Toronto. And I run a research lab, and on this picture you can see the lab and my kids and some of my lab members' families on our lab outing. So these are the people that are actually doing the work. I don't want to claim this work as mine. It's the work of lots of people working in my lab. And Rachel asked me to first say, why am I even working on NARS1? It's such a rare disorder. And the answer to that is because of Rachel and Terry. In 2023, I organized a conference on the NARS protein. So NARS minus the first letter N is ours, and that's a group of proteins that are all very similar. They all work in this construction site that Rachel just explained. And I work on several of the other ones, but I wasn't working on NARS. But after talking to Rachel, she said, well, there's just not a big community of researchers out there. We figured we could use our knowledge and just add NARS to our rooster of other NARS proteins that we already work on. And then if you want to go to the next slide, I can show what we're working on. So we have three different angles on how we approach this. So we work sort of on three different levels to study why the mutations in NARS cause disease, and then also try to find therapies that work with it. So the first level is protein. So we look at NARS itself and see how a mutation affects NARS itself. So this is really in a test tube, seeing does it behave differently. And sometimes this already will tell us a lot. So for different synthetase, we have found out that they're just not as stable as the regular NARS should be. And then when patients have fevers, that's when it gets really bad, because the protein is not as stable as it should be. And the fairly simple solution in that case was treat a fever right away. So those patients are very stable now because they get tons of ibuprofen as soon as they have the slightest fever coming on. So some things we can tell on the protein level. The next level we look at is in yeast. And that seems like a weird organism to work on because humans are not yeast. But those construction sites that Rachel mentioned, they are the exact same in yeast as they are in humans. So we can work in yeast as a very simple model system to study what happens to NARS or a mutation in NARS in a yeast cell and what does it do to the cell. So the advantage of this is that yeast is a lot cheaper and faster to work with than human cells or mice. So mice take 2 years to develop. My yeast take about 4 weeks. So that's why we work in a fast and small organism, because it gives you a lot of data. So I can look at all the mutations that you have found in your NARS community so far and tell you afterwards, it still takes time, but tell you what they do with the funding I have. And then finally, when we find something out, we go back to human cells and patient-derived cells. That's where the rare X really comes in helpful, because we otherwise have very little access to patient-derived cells. And we are very, very grateful for the support of the Rory Bell Foundation for the funding we got. And this helps us study the disease as we do. And it also helps us to get more data for more funding. So we're applying for a big Europe-wide grant right now with 6 other labs that also work on the RS proteins to further research in this area. And that's all.
I'm going to pause real quick to see if anybody has any questions for Ilka, or before we move on to Ingrid.
I think we'll get to it later as well in the like what we need from all of you section, but you heard Ilka talk a little bit about it. Is they do testing in other types of samples first, but then at the end of the day, they need to go back and test it on human samples. And so that's where we have been asking the community to donate those so that they can do the different drug testing on our patient samples too. And so I think that's just a really good example of where we've been asking people to donate blood samples and skin samples so they can do all the testing on that. And if we don't have those, then they have nothing to test on and make the case for that.
Yeah, I agree. For us as researchers, it's almost impossible to go further into clinical trials without showing something in human cells. So you can have all the evidence you want. If you don't show it works in patient cells, they're just going to stop you right there.
Thank you, Ilka. And then our second grantee is Ingrid Valley, who is at Scripps. And Ingrid is one of our young investigators. And actually, like, I just fortuitously met Ingrid in this conference, at this conference that Ilka mentioned in London, Canada, in line. And I had no idea she was working on NARS1, and she turned around and saw my badge, and she's, one of the impetuses for now why we have so many eyes on NARS1, because pure stroke of luck. So Ingrid, go ahead and take it away.
Hi. Yeah, thanks, Rachel. Yeah, as Rachel mentioned, it was really luck. I started working on this NARS1 project as part of my PhD thesis, and I had really no contact with the NARS1 Foundation, which I guess didn't exist back then. So it's been really great to be able to interact with Rachel and everybody in the foundation. Very grateful for the funding. So just a little bit about me. I'm from France. If there's anybody that has like the French families with the cases, I'm happy to answer any questions, also in English. But yeah, I'm studied pharmacy in Paris, where I was working on rare disease retinoblastoma, which is a rare disease cancer, and then decided to move on to a PhD to further complete my research training at Scripps Research in San Diego. So as I mentioned, the whole NARS1, what I call is NARS1 project, is my whole thesis, and I'm going to go a little more into it. Just want to mention that I'm working on it in Shanlei Yang's lab, where we focus on other synthetases. I'm specifically working on NARS, and then Leo, who's also here on the call, is working with me. Yeah, can you move on to the next slide, please? Right, so what do I actually do? So just like Ilka mentioned, we're working in test tubes, so what we call in vitro. So when you guys discover a new mutation, what we do is we extract the enzyme, and then we're going to study the mutation and what does it do to NARS1, and how can we explain basically how does it cause the disease. So I am mostly working on the 2 mutants, which are this arginine to stop variant, which is the de novo, so like the mutant that was not found in parents, but that's new in children. and this other mutant, which is the arginine to cysteine 545, which is the inherited and where both parents transmitted one allele. So the kids have both. And we're really, my question is trying to figure out what's going on at the enzyme level and why is it not working. And the goal of this is to figure out how we can drug this later on. And then Leo is working on trying to figure out how does NARS1 look like, and the goal for this is to be able to target it more specifically with drugs. Like when you have a visual of what something looks like, it's easier to fit something into it, for example. Yeah, just I have a little example on the next slide. Yeah, so it gives you, it's very sciencey, but it gives you an example of what NARS looks like. So that's a representation of what it would look like. And for those particular variants, the arginine to stop and the arginine to cysteine, they're on the right, they're in fuchsia and then bright green. And you can see that they're deep down in the little NARS1 pocket. And then when those are deleted, just completely removed in the case of the R534 star variant, you can see that it can cause a lot of damage. And also this other green one, you can see that, yeah, basically when it's removed and it's an important part of the architecture, so can cause collapse and a lot of problems. So that's what we're trying to figure out, basically.
Pause one more time for any questions. And I know that like this basically just looks like rainbow vomit to most of us, but this is
It does look like rainbow vomit.
yes, this is kind of like this is what this would look like all folded up inside our cells.
Right.
Okay.
And so this is how scientists look at this, in a way that they can understand it and see, because we can't look at that
Yeah.
at a cellular level otherwise. But why this is super important is because, with Ingrid's work and Stephanie kind of started this, she'll talk a little bit about her work here next, is different variants, right, can do different things to this protein. And so that rainbow vomit can look different, right, based on what's happening from that variant. And that can also change like what we can use drug-wise, because it could have a different adverse effect in one population versus another. So that's why this work is super important, because it really helps us understand, how this protein changes and what it's doing to other proteins around it. Hopefully that helps. If anybody has any questions, please shoot them now.
Can I share
I'll
my
stop sharing.
slides?
Yep. You go ahead.
Is our last grantee winner. You might actually recognize her name from one of the original publications. So Stephanie was actually part of the team that helped discover NARS1 as like causing, all of these symptoms and disorders. And so she was our single researcher a couple of years ago, and that's, really grown to a network now, thanks to, people like Ingrid and Ilka and others. So go ahead, Stephanie.
So hi everyone, I'm Stephanie. As Rachel said, I might have spoken to you already through different correspondence about your children and your mutations, but I'm basically a geneticist from London. I have degrees in biology and virology. And since 2015, I've been working at University College London at the neurogenetics lab, where I get to work a lot with directly with patients, but also other neurologists. And we look at the DNA of people that might present with different neurological disease. So we use a lot of sequencing. And as many of you might already be aware of, there's like different levels of sequencing, genome exome sequencing that you might have used to diagnose your children. But really my connection with NARS1 is because we've seen the first patients, two siblings from Pakistan with NARS1. We were the first ones to publish one of the two papers with the genetics of this disease back in 2020, where we presented the clinical features related to this disease. And since then, I've been very passionate about continuing this work. So I've been directly liaising with patients and other clinicians who might be seeing patients with NARS1. So we are still recruiting patients for further studying. I've been coordinating some of the collection of biomaterial from this family. So as Rachel and Terry have said, using patient-derived material is really important. I've been involved in a gene review paper. I've been working on a mouse model in the past few years, and I'm really keen to continue with raising awareness, applying for funding, and working on NARS1. So with the funding, very gratefully received from the Rory Bell Foundation, my proposal is about building on the cellular and molecular knowledge and understanding of why we get the NARS1 disease. So I have already collected some skin fibroblasts from patients, which I have already used for generating stem cells. For people that might not know what stem cells are, these are very valuable material that we can use to reprogram into any type of cell in the body that we like to study further. So I'm going to use the funding from the Rory Bell Foundation to use these stem cells to create specialized versions of neurons, which are the cells in your brains. And more specifically, we're going to use cortical neurons, which are the type of neurons that are responsible and that are really essential for the early development of the brain. And since we are talking about a disease that really presents quite early on in life, this is, we believe, the most appropriate model to use. So we will compare it with healthy controls, but also isogenic controls. These are basically patient lines which have had the mutation of NARS1 corrected, but still keeps the genetic background of that patient. And we will generate cortical neurons in culture, try to grow them for long enough to be able to come to some conclusions. And we will try to see what's different between patient lines and control lines. So we will use high resolution staining with different markers to see the proteins that might be active. We will try to quantify the NARS1 protein. We will carry out some enzyme activity assays to see how much of the NARS1 enzyme is still active. But most importantly, we will look at the electrical signals that these neurons might be producing in these cell lines. That is very similar to the signals that exist in human brains. And this all is very important because it can generate the knowledge. We can understand more about the disease with the aim of designing some therapeutic interventions. And this work will be carried out in our lab over the next 1 year and a half. And hopefully we will be in a better position to then have enough data to apply for more funding or bigger grants and to be able to plan some gene therapies. Yeah, that's everything from me.
How much would it cost for the gene therapies?
Sorry, how much is going to cost?
Oh, yeah, for gene therapy, if it does get to.
Yeah, it's going to be a big chunk of funding, so we need that preliminary data that we are hoping to get from this project.
Yeah, ultimately, gene therapy to get to a usable therapeutic is several $1 million.
Yeah, and time.
I have a question, please.
Yep.
A question, please. Is there any case or any example in other the mutations that gene therapy were found in such a short time?
You mean produced in a short time?
I mean, almost the same work in other mutations. Is there any work or any research similar to this one in other
And
mutations?
for NARS1 or for other
No, no,
disorders?
for other mutations.
Other mutations?
Other
Yes.
mutations or other diseases, do you mean?
Yeah.
Diseases. Sorry.
Diseases. Yeah.
Yeah.
Yes. Yes. There's a lot that's kind of in the pipeline with gene-based therapeutics. Not just gene therapy replacement, but there's other modalities that are being looked across several different disorders, both neurodevelopmental like NARS1 and like neurodegenerative disorders, which comes with a long regression over time. So yes, and there are FDA approved therapeutics for different disorders such as spinal muscular atrophy, Duchenne's muscular dystrophy. There's a couple out there for a few others too. So yep.
How about rare diseases like almost NARS1? Is there any nano-rare disease which has already gene therapy?
Not to our size, no. There definitely are groups that I'm in contact with who have therapeutics in the pipeline, either preclinical or starting some early phase ones. Their communities are, a little over 1,000 or so. But yeah, so there's definitely a lot of that's happening in this space very, very quickly.
Thank you.
Yep. Any other questions? Okay. So this slide actually is just a really nice summary that Kit built for us of, all of the things that we're collecting for this toolbox. And so you heard, Ilka and Ingrid and Stephanie talk about the things that they're doing. Some of that stems directly from trying to, facilitate some of this work, gathering samples from, patients around the world, building a mouse model, which, as Ilka already mentioned, is a much more complex model and takes a lot of time and energy and effort, but ultimately you need those to kind of test your therapeutics, your drugs in. Stephanie is developing those neuron models, which are key for us because of this being a neurological disease and other things. And so this is really just a nice summary and picture of where we are and how we're going to continue to move the research forward. Next slide. I think we're into, yes. So I really wanted to talk about RareX data. So Terry will talk about this too at the end and like how important and relevant this is. I know Stephanie has submitted to use this data. Kit and I have both presented RareX data at multiple conferences for scientists, for clinicians, so that they can really understand, what is happening on a day-to-day basis and what from your perspective, from the perspective of the caregiver. And the goal of RareX, which is a nonprofit, is really for you as a caregiver to be able to enter your data, your information from your child into this system. It's de-identified, but it really talks about all of the symptoms that your child has experienced over their lifespan. And it happens you fill out these surveys on every 6 months or on an annual basis. And it helps to build us an understanding of what NARS1 looks like, right, in your child over the span of their life. So this was our data from last year, which Kit actually presented a good bit of this at the RNA conference in Ontario this year. So we have 19 total individuals enrolled in RareX. And the main survey, which is head to toe, which is essentially tells you everything that's happened in your child's body over the course of their life through that survey has an almost 80% completion rate, which is really great. And then you also can upload your genetic report. And right now we have 10. So just over half. And so that's something we really need. Next slide, Terry,
And
please.
Rachel, if I can interrupt for
Yep.
just 1 second on this. As the non-scientist in the room, RareX is a very simple platform to use. It is surveys, and you can go through them and you can come back to them, and it doesn't you don't have to give up a week of your life immediately on it. You can register and you can go through the first couple of surveys and get the core information in there. And myself and Katrina just went backwards and forwards on it for a little while to get it up to date. If anybody requires any help or assistance or a little bit unsure, I've put my email address on the bottom of that slide, which I'm sure we'll share afterwards. I'm quite happy to set up one-to-one sessions to just take people through those initial stages of getting enrolled. It is really very, very important when we see the massive work that's being done by our different researchers. Is really, really important to have this done. And just as the kind of non-science person in the room, it isn't as daunting as it may seem when you hear about it. And I'm very happy to support people on a one-to-one basis if that helps you.
Thank you, Ross. That's very helpful because I too also just go in and try to update things. And most surveys don't take very long, maybe 15 or 20 minutes to do. I think for our families that have older individuals, that first head to toe can take a really long time because you're thinking back to your child from the moment they're born until their life now. And so I know I see the Andersons on here, so I know that they have participated in this and already. And I think the first one takes a really long time when you have a kiddo who's a young adult now. But this is we use this as a foundation. I know Stephanie has used this and accessed this data, and it's really helpful because we don't have a lot of publications, a lot of literature on NARS1. We're obviously hoping to change that by funding the research that we're funding. But until things get published, right, this tells the community, it tells the world, right, what your day-to-day life looks like and things that are missed or not included in publications because, that person writing that has only seen one kid, right? Or 2 kids or 10 kids. And it's very different. So I want to put a plug in for those who are already enrolled. And if you do enroll, please make sure that you upload your genetic report. This is super important for us because we can make correlations between variants and symptoms. And that really helps us to get a better picture of what's very common in some variants, what is not common, where there's overlap across variants, and really helps us think about clinical trials and outcomes that we want to change and how to change them. So this is how you do it when you log into RareX. The documents is on the right-hand side. You have to click it. And then upload and then select your tag for genetic report. So we only have 10, so just over half of the number of people enrolled. We need as many as we possibly can get so we can have a better understanding. Next slide, please.
Question on that?
Yep.
Rachel, this is Kathy Krebs. Being new to the organization, I'm not too familiar with that, but we're certainly very interested in sharing our information. Could you just tell us who has access to the data that we put in, who owns the data that we put in, and then ultimately what will happen to the data?
Yeah, great questions. I appreciate that, Kathy. Yeah. So all the data that goes in there, you have access to as the caregiver. So you can see your data, your information, and then it gets de-identified. So on the back side, right, they strip they strip away anything that's like, your child's name, your child's birth date, information like that would identify, right, who your child is. And then the only so for instance, Stephanie accessed, our RareX data for looking at this as part of some other natural history work. She just gets a data set with a bunch of letters and numbers, but tells the story of symptoms. So that survey data gets put in, and so we can see that, but we don't know who your child is. And so it's all de-identified. You own the data. So you as the individual, at any given point can pull your data out, can unconsent, it gets erased, it gets deleted, never to be seen again. But hopefully that helps answer your question. And access wise, we access it. The Roy Bell Foundation accesses this once or twice a year to help to build some graphs or to get a better understanding of symptoms across the population, and it helps us to present that. Again, we don't know who we're looking at. We just have an aggregate data set, and sometimes actually they'll just make the graphs for us, so we don't have to pour over data ourselves. So is that helpful? Does that answer your question? And then researchers can obviously do the same thing.
Now, you say researchers, is that only limited to our researchers that we're
Anybody.
talking to here, or anybody?
Yep.
So it's
Yep.
on a database that anybody can access.
Anybody can access So
if I may chime in, it's really hard as a researcher, at least from Canada, to get access. You have to go and have your institution sign off on you.
Yep.
It's not any student who wants to look. So
Correct.
I so far have not gotten access because it's still sitting with our legal department because they have to sign off that Western vouches for me. I don't know how it was for Stephanie, but it's
Same.
really so it's
It
not
took me
any
more than a
random
year.
it's not any random researcher that can look at it. It's a really, you have to have ethics approval and stuff like that.
Yes, thank you, Ilka. Yeah, so yeah, I think it took Stephanie, gosh, we probably went back and forth for a year, maybe.
Yeah,
Yeah.
that's what I said, a year.
Yeah. So, right, you have to have an IRB, you have to have sign off with your institution. And so any researcher can submit for that, but that is not going to be released without the appropriate use agreements and infrastructure and protections in place.
Okay.
Taking the big view, I just want to step in and say that RareX is a subsidiary of a very well-known nonprofit. It's a nonprofit itself in the rare disease space called Global Genes. And so it's owned by the nonprofit. It has all sorts of HIPAA compliance and European and international equivalents of data security. So we feel very comfortable about it from that perspective as well. I just wanted to make sure that got mentioned.
Yeah, thank you, Kit. Yep. Yeah, it was a big reason why we went with them is also because they support the international community and have a lot of the privacy agreements already in place. And so, that obviously for us is very important to protect your privacy and in our community's privacy and your right to own your own data, which is also not always the case with other registries of this kind. Any other questions?
Thank you.
Yep, great. Thank you. Great questions. So this is just a really high level of the RareX data that we have. And so as I mentioned, we had 19 individuals enrolled in 2024, yet we only have data for 15 individuals. So that actually means we have 4 individuals that haven't really completed any surveys. They've just enrolled. So again, it's so important, to be counted, to be included in this aggregate data, to be part of these data sets, that these surveys are done or some surveys are done, because otherwise we don't get any information. I've put here on my left-hand side, on the left-hand side, sort of what I think about as priority surveys, or what we would in terms of NARS1 and how it's going to affect the systems. So head to toe, most important, like I said, And then what happens is you get generated these level 2 system surveys. So, right, if you click, like there's global developmental delay, then you'll get like a brain and nervous system survey to complete. And that just gives you a better idea of cataloging your child's journey in terms of their symptoms. And then I've put in sort of other sub-surveys. These ones are very quick, minus the Vineland, that are, pediatric eating system assessments. So we see quite a bit of GI issues. And so this is a really important, survey for us to think about, right? How that's impacting your kids. The PELS survey, which is for epilepsy, which would only get dropped if you have checked that your child has had seizures. Quality of life. This is something that is not measured very often, but we know that it's obviously very impactful on us as parents. The ORCA, which is communication. And this is really key because it is actually being tested in a lot of individuals and disorders like NARS1, where communication is so significantly impacted, in the ability of individuals with NARS1 to, have a lot of, be able to communicate verbally, be able to string a lot of words together in sentences. And so this is a really good survey for that. Sleep, Vineland 3, Vineland, I will just say it is brutal. It is easily a 2-hour survey. Do not do it in one sitting. Do it in multiple sittings. But it covers a really broad range of symptoms, development, behavior, communication, and is used in clinical trials. And so when you have a survey that's used in clinical trials where you're looking for changes in those measures, they're really important that we understand baseline so we can understand like how drugs, how therapeutics can change that. And then the last two are is a behavior and a development survey, super fast. Next slide, please. And then again, I just wanted to show you what this looks like. Like when we look at head to toe, so of the 15 families that we've had, do the head to toe survey, and this would be last year and technically the year before because the head to toe is every single year. So you can catalog new symptoms. You really see like how NARS1 is impacting individuals, across the body systems. And then I know we're getting fairly close to time. So the next few slides are really just for you to kind of look through yourself and to try to see, a little bit more insight into how we get information or how the researchers, right, like Stephanie or Ilka will get information and can be able to graph it in a way. So this is a breakdown specifically of the brain and the nervous system. And that teal color is, the number of individuals that are reporting yes, right across the system, across your nervous system. And again, it helps us to see how this is impacting, kids across their individual body systems. And then I'll just skip ahead. Honestly, the rest of the slides look very much like this. Terry, if you want to go to yours section.
Oh, I was gonna say, I blocked 90 minutes for this. Can you hear me?
Mm-hmm.
Oh, we have 90 minutes. So if there's any things you want to cover through, like quickly, go ahead.
Go to the next one real quick because I think this is GI. Yep. And so I like this. I like this slide because, this was something that really the GI system, the digestive system, and the impact that NARS1 has on it is not very well reported in the literature. And so this is an example of where your data, your information, your story really makes a huge difference because this helps tell like what is happening every single day that isn't being reported. And it gives opportunities for clinicians and researchers to really understand how diffuse, NARS1 is. And so we know from this, like, that NARS1 kids like are likely to have feeding difficulties and constipation and, be smaller. And so those are all very, very helpful pieces of information that can help, inform clinicians and researchers something that maybe they aren't seeing or didn't know, especially in a disorder that is so rare. There's
Rachel,
one
there's a
question.
question in the chat from Rosalind about testing asparagine supplements. Do you want to speak to that at the end or no?
Yeah, we'll do it at the end. Yep.
Okay, we'll come back
Okay.
to that.
Yep, and you can just go ahead. Again, these are very similar. I just wanted to give some examples.
Okay, so then switching over to this year, things like non-research related, we have set forth just a couple of different goals in terms of where we want to grow, how we want to service you all better too. So first of all, we don't operate without fundraising. So hoping to grow our fundraising efforts. We have a couple of things that we've penciled in on the books for the year. So the first one being a worldwide 5K. So the Boston Marathon is a big deal here. Rachel and her son and a few others are running a 5K that weekend. And our event platforms that we have allow for peer-to-peer and now some international capabilities. So we can all try to do the similar thing, but maybe a 5K from our own homes around the same time. So we'll be working on that and working to just communicate out how people can join on that. And if you want to tag on, if you want to support that in any way. But we know that having both US and international capabilities are really important. We have also been really successful in hosting an in-person event every fall. Now we've been able to raise quite a bit of money that way. So this year our event will be in Detroit in September. So for anyone who is local in the US or wants to travel in to visit that, that's something that we're starting to think about planning for now with one of the other families in the group. Andrea, she's not on the call here today, but they live in Detroit, Michigan in the United States. Then also just hosting some in-person family and research meetings. So just to be able to meet all of you wherever you're at. So we know we have a pretty widespread population, as you saw on the map, some US, some South America, a lot in Europe as well. So for anyone who's around the weekend of the Detroit meeting, we're hoping to have a family meetup for people who are there. Rachel is going over to London in March, and I know she'll be seeing Stephanie too. So we'll spread the word for anyone who is local in that area to stop by that day. I believe they're going to visit the lab and just do some sort of a small meetup there. And then in September,
Can I say something real quick about London, Terry?
Yep.
Yeah, just this is where my son and I are spending our spring break over there. And we're gonna go hang out with Ross some too. But if you guys are in Ireland or the UK or like have interest, like please let myself and Ross know. So we would like to coordinate with Stephanie. She's graciously allowed us to perhaps be able to do a lab tour and other things, either that Monday or Tuesday of our spring break, which is March 24th or 25th. Thank you. That's it.
That's perfect. The last one is the ARS conference, which you heard Ilka talk about. So it's every other year. That's where we were able to meet Ilka. We ran into Ingrid as well. We're able to kind of present, meet lots of other people. That is going to be in Croatia the last week of September. And so we will be trying to organize something the weekend prior at that location for anyone who is over in Europe too. So more details to come on all of these, and we'll make sure to share with you how you could get involved and what the options are for you. The other thing is quarterly newsletters. So some of you might have seen these, but we started doing them last year just to share out more of what we're doing. We typically post them on our Facebook group too, so you can join the listserv for that, but just trying to get the word out a little bit more often on what we're working on and what we're doing. virtual family calls just like this. So I think we've been only having maybe one-ish a year, possibly two, but I think we'd like to make sure we're having at least a couple of them every year, just so we can invite all the new people in and then keep people updated with what we're working on. To that point, focus on family engagement. So Ross has created a WhatsApp community too. I know there's a lot of people in our Facebook group and a lot of different questions that people have, especially when they first join, wanting to know other people in their situation. And so he's created this community. If you want to join, the QR code is on the screen, but we'll also share it out with different focus like topics of groups that you can join if you want to, or you don't have to, so that you can talk and share stories with other people. Anything you'd
If I
add
could
on that,
just say,
Ross? Yeah.
yeah, just on that. Look, this is an idea, and I hope it's something that proves to be a good idea. I guess on these calls we talk an awful lot and we share a lot, and we're keen to create a space where people could share on different topics. The Facebook group is great, but things kind of get lost over time a little bit, and questions resurface again. So by creating the WhatsApp community, the idea is that we can create different kind of chat threads that people can talk about different aspects, and also as a way maybe just for the families to connect and get to know each other. It is all open for debate. So drop me a message if you feel like there is a particular kind of a chat topic that people would like to see, or if you wanted something country or regionally specific, or any topics. We'll obviously keep an eye on it, and if we think that there's a better structure, we'll put it in place. But it is there for you to use as a resource and to share information with each other and to just build that engagement across the families. Because we appreciate there isn't an opportunity when we're talking to you like this for an hour and a half, for some of that cross communication to happen. So any way we can facilitate it, we're always open to your feedback. We're all learning a little bit here too, so don't feel like we can only do it the way that we say, right? We need your feedback on how we make the stuff useful.
Awesome. And then the last one is just working on technology a bit more, right? We have done a lot of things manually as a foundation, and spreadsheets and sheets and forms. And so with the grant as well, it's kind of allowed us to invest in some new technology and platforms and just applications that allow us to more streamline things and capacity build. Okay, so for all of you, how can you help? I think we already talked a lot about Rare X, so we can't give the insights to the researchers if we don't know all the information. So I think we learned a lot from our first round of Rare X, even with 10 people, of there's digestive issues. Oh my gosh, we all have that. There's sleep problems. Oh my gosh, we all have that. That's not printed in the literature anywhere. So I think it's just so valuable to be able to share all those insights, and that's where we're getting all the information from, is the surveys from all of you. The second one is fundraising. So we can't do what we do, and we'll continue to fundraise and host events and try to get people engaged as much as we can. But we've had a lot of success with people all over the world doing simple things like car washes, golf tournaments. It's really inspiring some people, Kat and Ross, to get in shape and run some marathons and half marathons. And we had a team here in Detroit do that last year too. So no opportunity is too big or small, and we do have the ability now to do online things internationally. And so if you ever have ideas, shoot our way. We're happy to help and facilitate in any way that we can, and we'll make sure to spread the word on any things that we're doing that you can tag on to or help out with. The third one we talked a little bit about earlier too, when Elko was talking about providing your samples. So I think we're always trying to make sure if there's an opportunity we're in the area or out doing blood collection or sample collections. I know Rachel used to do quite a few of these, but we can't do our testing to make therapeutics unless we can test them on our own human cells. And so if that opportunity arises and it's near your area, we would definitely ask that you try to participate in that so we can build even more cell lines and iPSCs and testing on things. Anything you'd add to that, Rachel?
No, I think right now I would just add like from a foundation perspective, we have our own biorepository here in the US that I can only collect for in the US. However, we also have like Stephanie mentioned, we've been able to work with her and UCL to get other samples, and then I've been Ilka is starting to connect me with some other clinicians and researchers in Europe. And so I think there'll be opportunities to be able to expand some of those, especially to get as many different variants that we can. So again, we can have a better understanding of, how they might be different or similar.
The next one is just have your clinicians engage with us. So the more doctors we know around the world who are having NARS1 patients that they're working on, the more we can try to connect them to share stories and link them together and start to create a group of clinicians around that. There's very, very few of them. And so I think that they can learn a lot from us and also from each other. Next, share your expertise. So this is kind of how our board functions. We each have an area of expertise, and if you're good at something, we will definitely take your help with it. So Kat, I will call you out again, but she totally helped revamp our website. We have, people who raise their hand and volunteer with different areas of expertise they can help with, whether it be social media, financials, things like that. So if you have expertise and there's something you feel like you could add value or want to help with, reach out to us and we would love to have you. And then the last one, does anyone have a question?
Can you put in the chat the WhatsApp information?
Yes, I will do that now.
Okay, thank you.
Yep. Okay, and then the last one is just promoting NARS1. So promote NARS1, the foundation, when you're ready. We know sometimes people aren't at a place that they are ready to share with the world or speak about it yet, but when you are, in terms of other rare diseases within your community and gathering support for different organizations, it just helps so much for us to build our network. And the more people that know about us, the more people that can help. Any questions on any of this before we move on? Okay.
I'll just add one thing. Terry, I think Kit popped this into the chat, but for some of our families, especially in London, and if we're thinking about, we really do want to do something with Croatia up against the Rs meeting, is please just make sure you fill out this JOT form, which is, again, it's privacy protected, but that way we have your contact information so that we know how to reach you via email so we can set things up.
Okay, and then the last thing we have is just resources for all of you. So Ross has put together some really great resources. I know things like us as parents like wish we would have had when we first got a diagnosis and we're trying to navigate things. And then just a couple of other informational. So our website, I don't know if you want to drop it in the chat, Kit as well, but we just launched our new website this week, so it's been completely revamped. Should be a lot more user-friendly for everybody. Take a look at it and let us know what you think. So that's in there. The site name hasn't changed, just has a new look. And then we have our Facebook page. Most of you are part of the private group that we have. We have an Instagram and a YouTube channel that we post videos that we've made for some of our events with a bunch of your kids with your permission in it. So those are pretty fun to see. And then the bottom section is just, we have a few families that have their own pages for their kids that you can follow along with too. So when we send these out, they're actually like links that you can click on. So would love to add yours too if you guys have any. That's all we have, and miraculously still have some time left that we would love to open for question and answers. I think the first one we could start with, Rachel, is just about the asparagine testing.
Yeah, and I'm going to let Ilka just give an update on that since she's been leading some efforts and charges around testing, and yeah, take it away, Ilka.
Okay, so to first of all, I'm a researcher, I'm not a physician. So just to clarify that I only see this from the scientific point, but I do work with lots of physicians. So the NARS is one of 20 different proteins that do a similar job. They are specific for one amino acid each, and NARS is for asparagine. So for some of the diseases with the other synthetases, we have had really good success with amino acid supplementation, where that can help stabilize patients or even improve patients. We have this has not been tried for NARS, okay? But it has been tried for 5 or 6 others. And there are centers, and there's one in the Netherlands, and there's one in France where they are doing this, and we have one here in Canada where we're doing this. In my lab, with the great funding we just got, we have tried this in cells with asparagine to try this just out in our test tubes, and it has worked. We are now trying this with patient-derived samples. So as I explained before, when we do our research, we go in simple models first, and then we go in your patient samples, because the patient samples are different from what we have in our lab, where we try things just in lab-grown cells. So if this is successful in those patient samples, which will take a little bit to try out, we do have physicians that are willing to try it with asparagine. So I have connected Rachel with the physicians in France and the Netherlands who know a lot about these RS diseases and who are willing to help with that. And we have one here in Canada. I don't know if there's other physicians who are willing to try this. So the nice thing about this is it's a natural, asparagine is a natural amino acid. So this is not a real drug that you need a clinical trial for. So it is very promising. It might not work for all mutations. It might work for some. So we have to be cautious and being how hopeful we can be. But our general idea is it's likely not going to hurt. So we are working on that. And if once we have more updates, I will always keep Rachel in the loop and send out whatever data we have, but we are working on it, and we're actively trying to find a cure here. And asparagine supplementation is one of the options we're really getting data on, and we're getting good cure. Or good data for in our test tubes. So we have one cell model right now with the 534 stop mutation, and we will try to get more patient samples so we can look at more different samples to see if it works or not. So that's just the update I wanted to get. But if you want to be connected to any of those physicians, Rachel has all that information. And
Yeah.
also, physicians are a great resource. They treat other patients with these diseases, so they might know more than your regular physician about these diseases.
Yep, and I think Ilka, we have them on the books, I think soon. I just haven't sent out an invite yet, but yeah, so I think we'll have more information out after that in terms of like, you know, the best way to connect, what their thoughts are. So, you know, when I think about using supplements or thinking about drugs in general, right, we want to think about like measuring how that might impact your child and what might, you know, be beneficial, what might change as a result of that. And so this is part of, you know, clinical trials, but doing it in a very thoughtful way so that once you have that data, once you have that information, it can become publishable. It becomes part of standard of care. It becomes, you know, recommended that, you know, for every kid that gets, you know, diagnosed with NARS1 disorder, like you can try this and at this dose, and this is what we may expect. And just, you know, Ilka mentioned this, it is a supplement, it is over the counter. You have to be very cautious with supplements because it is not regulated. And so because they're not regulated, you actually don't know what's in them. And so there's very key certification processes and manufacturers that I look for as a pharmacist, as a background, that, you know, so that's part of this is, you know, kind of looking to see, you know, what is available. Is it available as a liquid, right? What's a good dose that we might need to look at, other published literature. So it's not as simple as just going and buying it and giving it to your kid. We try to do it with a little bit of guidance and evidence, but we are very hopeful about you know, this as a possibility. Any questions?
The supplement
What
that you,
is it?
oh, sorry.
Oh, go ahead, Roxana.
The supplement that you're talking about, the asparagine supplement,
Yep.
what results, if any, do you know that it helps with?
Yeah.
There isn't.
Yeah, so we wouldn't know. So like what Ilka's doing right now is testing it in cells, right?
Yeah.
And so cells, right, and yeast models and ultimately human models. And what we won't know, like, right, those cells don't talk, right? They don't walk, they don't write. So what she's looking at is just a shift or a change
in anything.
In anything, yes.
Yeah.
And so when I think about that in terms of people, right, in humans, right, that's where some of this thought around rare X is super important, because I think about like, what's the most important thing to you, Roxana? Like, what is right, right? And so maybe it is, maybe Joseph says like, 4 more words, right? Or you have a little bit better understanding in terms of his enunciation, right? Maybe for some of these kids, it's, they go from walking to running. It's like, so it might be a little bit different for everybody.
Yes.
But those
roles. I mean, I know with Joseph, there's certain aspects that not have changed for years because he's obviously 27 in March. But there are bits that have got worse. His eating, for instance, and things like that. And I don't want him to lose that. I'd really like to keep that. He has got a peg tube and stuff, but that obviously impacts his speech as well if he's not eating.
Yes. So those will all be things that I think, I'm very particularly interested in trying to kind of devise, right? And how we figure out how we measure those changes, because ultimately that is not just good for your child, right? It's good for, any child moving forward, that gets diagnosed. And so hopefully with those conversations with, Ilka's connections and some of the clinicians that I've worked with here in the States, we can come up with something.
If I can chime in. So what we've done for our HARS patients, so these are histidine instead of asparagine. This natural history, we call that, is what you fill out on Rare X, where we know how the disease progresses. If we know that, then we compare that to what we think should happen. So we have this for histidine. We had lots of patients. We had 20 patients in our trial, and we knew the children should, in principle, all go deaf and blind because that's what they've always done.
Right.
And the histidine do not go deaf and blind. They're totally fine. And they weigh more than their peers, right? We know the weight of the comparator group, and then these kids, they have more normal weight. So having this history in the Rare X will really help us establish a natural history. In general, for some amino acids, we did see improvement of the condition, but in general, it's probably more a stabilization and not getting worse. That's our thought. It's like mine as a researcher. It's hard to undo damage that's done, but you can prevent more damage. So that's our thought. But for methionine, we did see improvement in France. Those are cases in France. So it's hard to predict how it's going to be with NARS and if it's going to work. But it's promising. So it's something to really consider, I think.
Excellent. Thank you.
Yeah. And I think about, I think that's one thing that Ilka's doing, and, ultimately, well, with Stephanie too, is, asparagine is not right, the end all to be all. We don't even know, but right, there might be multiple different, therapeutics, right, that you might need to put together to kind of maintain some of that, right? Or to, maybe, have some progress or shifts or changes. And those are all things that we're hoping to gain a little bit more knowledge and insight on here in the next, year with some of these grants.
Brilliant. Thank you.
Yeah.
What other questions do you all have?
I have a question based on something you had mentioned early on. Charcot-Marie-Tooth disease. It was mentioned. That's the first time I ever heard that NARS was linked in any way to Charcot-Marie-Tooth. And the reason I asked that is because Paul was diagnosed originally as a possible Charcot-Marie tooth. However, where the irregularity was not associated with Charcot-Marie tooth, but it was one of those, well, there's not enough research out there. Let's wait and see. And then about a year and a half ago, they said, no, it's definitely not Charcot-Marie tooth. That difference was not associated with Charcot-Marie tooth. So now they're saying you definitely have NARS1. So now if there is a link, is it you have both?
Yeah,
Yeah.
it's a great question, Kathy. So a couple of things. So Charcot-Marie tooth is very common in a lot of these other RS1 genes that like, ILCA studies, Scripps studies, and so where it's peripheral, right? The neuropathy part. And we have Rob Burgess, who also studies a lot of this huge cluster of RS genes, published a paper on NARS1 related Charcot-Marie tooth.
They do. All right. Yeah.
And he's also helping us with our mouse models here or at Jackson Labs too. So we know that it is also causing the peripheral disorder. We also know from some of the work that Stephanie initially did with characterizing NARS1 as the neurodevelopmental disorder or the neurological disorder, that peripheral neuropathy, that Charcot-Marie tooth symptoms shows up in kids. We also know that from talking to a lot of our families that peripheral neuropathy is part of the NARS1 symptoms. And so it's both. So
Yeah.
if you have NARS1 with the neurological, right, global developmental delay, it's very likely that your child also has the peripheral neuropathy as well that goes along with it, unfortunately.
That's interesting because I'd not heard the same as you, Kathy. We were told Joseph had Charcot-Marie tooth and didn't have cerebral palsy, and then he wasn't diagnosed till he was 22, and then he didn't have Charcot-Marie tooth anymore. He didn't have cerebral palsy anymore, but nobody else that you see knows any more about it. So, but that's interesting because I hadn't heard Charcot-Marie tooth linked with it before until you said
it's
it
brand
today.
new. Yeah, it's brand new, just published last year. And since then there's been there's two total publications out now on it, on NARS1, causing it.
So the peripheral neuropathy portion of it is the one that's associated with the Charcot-Marie tooth. Is that what you're saying?
Yep.
Okay.
Yep.
And it's funny because, so like, Roxanne, like your son, Paul had first diagnosis that was incorrect or may not be incorrect. I don't even know at this point, cerebral palsy. But then when the CMT came along, oh, well, it's CMT because cerebral palsy never got worse. Well, now they're saying, oh, yeah, it can get worse. But the CMT never dealt with the learning disability portion, which always made that stick out in my mind that maybe this is not a correct diagnosis.
No, I think the Charcot-Marie tooth is more physical, isn't it? It's more, not mental or represented in that way. It's more physically not being able to walk. I mean, they wanted at one stage they wanted to amputate Joseph's legs because he had Charcot-Marie tooth, and they said they're never going to get any better. And I'm really glad they didn't because
that piece can have the pain from it, the symptoms, can certainly impact sort of, your mental state, and how you're able to function and able to keep up and do things, because it can be so significant. But yes, what we see in, more of the vast majority of our NARS1 population starts with the global developmental delay, starts with the missed milestone, starts with all of that. And this is an added bonus, unfortunately. And I think Roxana and Kelly, like you guys and James's parents too, you guys are our trailblazers and got a whole lot of wrong diagnoses, I think.
Yeah. Yeah.
Before we got to here.
Yeah.
So we certainly appreciate that and hearing from you and
Oh, well, I know
sharing.
when they mentioned Charcot-Marie Tooth, obviously you Google the life out of everything. And whilst he had the physical problems of Charcot-Marie Tooth, he didn't have, most people that have it are in incredible pain. Well, Joe has a really high pain threshold. He wasn't in pain. And most people elect to have amputations because they just want to get on with their life. Well, that was different for Joseph, and we didn't ever really get on board with that. So yeah, that's something we ruled out quite early, that he wasn't a typical Charcot-Marie Tooth.
Did you have a diagnosis, Roxana, that that was actually specifically on that gene for CMT?
No,
No.
they did some tests, and in London they said that he had Charcot-Marie Tooth, but I guess it is a variation of the NARS1.
Yep, and they wouldn't have been able to find it, right? Because
it didn't
Stephanie's
exist.
team didn't, they didn't know about it until just a few years ago. So
yeah. Because Paul's 36 now. And so when they said CMT to us, that was middle school back then. And luckily he has not had any dramatic changes physically or intellectually. So we're very, very lucky. And his pain tolerance, either it's very, very high or it's just not as bad. So he's lucky in that portion too. But there, I'm sure there are things that are there.
Stephanie, can I ask you a science question? Actually, any of the scientists, how do you measure can you measure different pain thresholds when that is generally what we would call, right, a patient reported measure? Because I feel like this is something I hear a lot or see a lot or feel happens in this population.
Yeah, I mean, there's an assessment that clinicians usually do for like a pain threshold. I can get more information from Professor Holden if that would help actually, and to guide you. But one thing that I wanted to mention about the CMT that you've been discussing, like CMT was already kind of linked from the initial study. It's just there was a different subtype of CMT, like the CMT1 and CMT2. One of them has to do with like the differences between which part of the nerve is impacted. Is it the axons, the long part of the nerves, or is it the myelin, the thing that covers your nerves together? So we had already reported CMT myelination issues with our patients. The paper from Robert Burgess was that he was reporting axonal neuropathy, which was completely new. And from a clinical perspective, there's like very very clear differences between CMT subtypes when they see it in clinic. So that was the novelty for their paper. So I'm not surprised that some of you were already seeing CMT in your diagnosis over the years, because probably your children have done like nerve conduction studies, which is the CMT test that everybody does to see whether they have a neuropathy issue. But regarding the pain, I can find more details about it and get back to you. I'm not entirely sure how they do it in clinic, but there is an assessment.
I think it's just I don't know at what point, right, some of these individuals would actually signal, right, pain based on what I know about the kids that I've met or watched them, because I can tell you, like, I've watched Grady like walk on his ankles and I'm like, oh good God. It makes me hurt, but he's like totally fine. And so
we see as well
Same
with
thing with
Kean that sometimes if he has a fall, there's almost a delay. He'll fall and you're like, you're waiting because you're like, it's going to happen in 2 or 3 seconds, and then it's like it builds to arrive. But it's almost like the pain has to get to where we'd feel it and it would build. They
Immediately.
don't feel
Yeah.
it until it's already built. It kind of feels like that, so it kind of comes across as almost a delay.
Yeah.
I am conscious of time, and I'm going to be really greedy because I want to do one thing. We have really great engagement, and just for our own wares, if it would be great if whoever can come on video for a second did, because it would be great just to get a snapshot of the meeting and all of our families that we have on here. Lots of people on video. Because we do have people disappearing all the time. I know I've done this in the middle of a great conversation, by the way, so apologies.
You just want a selfie, we know.
This is for a cat to put onto the website or something, I'm sure. We won't do that, don't worry. Everybody smile, 3, 2, 1, give each other a wave. Oh, I've lost that. Hang on, here we go. Oh, there's still people turning on their camera. Look at this. What a response.
Later response.
Okay, if you're not in the next 3 seconds, you're going to have missed the moment because I'm not going to hold this up any longer. And done. Thank you, everybody.
Thanks, Ross.
Thank you. Thank you, Ross.
Terry or Rachel, do one of you want to wrap us up?
Yeah, I think thanks everyone for joining. We appreciate the time. I hope, please let us know. We tried to get you up to date with everything that is happening, what we're working on, let you meet the people behind the scenes that you're always getting messages from and asking you to do things. Don't be a stranger. Feel free to reach out to us. We will make sure we share this recording with the group and the deck that we went over so you have some of the links. And again, reach out to Ross if you have questions on the Rare X and how to get up to date. We do have a ton of documents that we can share and a video on how to go through it as well. But all of us are here to help and we are all in it together. And yeah, we will try to make these more frequent for you.
Thank you for your help.
Thanks all.
Thank you,
Take care.
everybody.
Thank you.
Thank
you.
Bye.
Bye. Bye.
Thanks all.
Bye.
Take care.
Thank you.
Bye. Thank you.